Auxins are a remarkable class of plant hormones that have captivated the attention of botanists, molecular biologists, and agricultural scientists for more than a century. Their discovery and subsequent characterization have provided profound insights into how plants regulate their growth, development, and responses to environmental stimuli. The term auxin is derived from the Greek word auxin, meaning to grow, which aptly reflects its central role in orchestrating plant growth processes. The most well-studied natural auxin is indole 3-acetic acid, IAA, but the auxin family encompasses a range of structurally similar compounds, both natural and synthetic, that share the ability to modulate plant physiology. Understanding auxin biology is not only fundamental to basic plant science, but also has far-reaching implications for agriculture, horticulture, and biotechnology. The story of auxin begins in the late 19th and early 20th centuries, with pioneering experiments on phototropism, the directional growth of plants in response to light. Charles Darwin and his son Francis, through their classical experiments with grass coloptiles, observed that the tip of the shoot was responsible for perceiving light and transmitting a signal that caused the plant to bend towards it. This led to the hypothesis of a growth-promoting substance that could move from the tip to the elongating region. Subsequent work by Dutch scientist Fritz Wendt in the 1920s resulted in the isolation of this substance, which he called auxin. Wendt's experiments, including the famous oat coloptile curvature test, laid the groundwork for the identification of auxin as a mobile chemical regulator of plant growth. At the molecular level, auxins are characterized by their small size and chemical versatility. Indole 3-acetic acid, the principal natural auxin, is synthesized primarily from the amino acid tryptophan through several biosynthetic pathways. Alternative routes, such as the tryptamine pathway and the indole 3-pyruvic acid pathway, reflect the metabolic flexibility plants possess to maintain auxin homeostasis under varying physiological conditions. Beyond IAA, other naturally occurring auxins include phenylacetic acid, PAA, and indole 3-butyric acid, IBA, each with distinct roles and distributions within the plant kingdom. Synthetic auxins, such as 2,4-dichlorophenoxyacetic acid, 2,14-D, and naphthaliniacetic acid, NAAA, have been widely used in agriculture as herbicides, rooting agents, and tools for plant tissue culture, further highlighting the practical significance of auxin research. The distribution of auxin within plant tissues is highly dynamic and tightly regulated. Auxin biosynthesis occurs predominantly in young, actively growing tissues such as shoot apices, young leaves, and developing seeds. From these sites of synthesis, auxin is transported throughout the plant via specialized mechanisms. The polar transport of auxin, a process dependent on specific transporter proteins, ensures the directional flow of auxin from cell to cell, often from the shoot apex downward toward the root. This polar auxin transport is mediated by pin-formed, efflux carriers, and auxin-resistant, like AUX1, LAX. Influx carriers, whose asymmetric localization in the plasma membrane determines the directionality of auxin movement. The establishment of auxin gradients through these transport processes is crucial for the spatial and temporal regulation of developmental events. Auxin's influence on plant growth is multifaceted, affecting cell elongation, division, and differentiation. One of the classic actions of auxin is its ability to promote cell elongation in stems. According to the acid growth hypothesis, auxin stimulates the activity of proton pumps in the plasma membrane, leading to acidification of the cell wall. This decrease in pH activates expansin proteins and other cell wall loosening enzymes, allowing the cell wall to stretch and the cell to expand. In roots, however, high concentrations of auxin can inhibit elongation, illustrating the context-dependent nature of auxin responses. The hormone also plays pivotal roles in the formation and maintenance of meristems, the induction of lateral and adventitious roots, vascular tissue differentiation, and the regulation of fruit development and abscission. At the genetic level, auxin exerts its effects by modulating the expression of a large array of target genes. The core of auxin signaling involves the perception of auxin by the transport inhibitor response. Auxin signaling F-box, TIR1-AFB, family of F-box proteins, which function as auxin receptors. In the presence of auxin, TIR1-AFB proteins facilitate the ubiquitination and subsequent proteasomal degradation of auxin, indole 3-acetic acid, AUX/IAA, transcriptional repressors. This degradation releases the inhibition on Auxin response factor, ARF, transcription factors, allowing them to activate or repress the transcription of auxin-responsive genes. The complexity of this signaling network is increased by the diversity of ARFs and AUX/IAAs, the presence of feedback loops, and the integration of auxin signaling with other hormonal and environmental cues. Auxin's role in pattern formation and organogenesis is particularly striking during embryogenesis and post-embryonic development. During embryonic patterning, localized auxin maxima established by the coordinated action of biosynthesis and polar transport direct the formation of the apical basal axis and the specification of embryonic organs. In the shoot apical meristem, periodic accumulation of auxin at specific sites triggers the initiation of new leaves and flowers, a process known as phyllotaxis. Similarly, in roots, local auxin accumulation specifies the sites of lateral root initiation. These developmental processes underscore the importance of precise spatial and temporal control of auxin distribution and signaling. The interplay between auxin and other plant hormones adds another layer of regulatory complexity. Auxin interacts synergistically or antagonistically with cytokinin, gibberellin, ethylene, abscisic acid, brassinosteroids, and strigolactones to fine-tune plant growth and development. For example, auxin and cytokinin have opposing roles in the regulation of root and shoot meristem activity, while auxin and gibberellin cooperate to promote stem elongation. The crosstalk between auxin and ethylene modulates responses to abiotic stress and controls processes such as root hair formation and fruit ripening. These interactions enable plants to integrate internal developmental programs with external environmental signals, optimizing their growth and survival. Environmental factors profoundly influence auxin biology. Light, gravity, temperature, water availability, and nutrient status all affect auxin synthesis, transport, and signaling. Phototropism and gravitropism, two classic examples of plant adaptive responses, are mediated by differential auxin distribution. In phototropism, exposure to unilateral light causes redistribution of auxin to the shaded side of the stem, promoting cell elongation and bending toward the light source. In gravitropism, the perception of gravity by specialized cells in the root cap leads to asymmetric auxin accumulation, resulting in differential growth and root curvature. Stress conditions such as drought, salinity, and pathogen attack can also modulate auxin levels and sensitivity, influencing plant architecture and defense responses. The agricultural applications of auxin biology are extensive and diverse. Synthetic auxins have been employed as herbicides, particularly for the selective control of broadleaf weeds in cereal crops. They are also used to induce rooting in cuttings, promote fruit set and development, delay fruit abscission, and manipulate plant architecture for improved yield and quality. Advances in our understanding of auxin biosynthesis, transport, and signaling have opened new avenues for crop improvement through genetic engineering and molecular breeding. For instance, manipulating auxin-related genes can enhance root system architecture, increase stress tolerance, and optimize plant stature for high-density planting. Recent years have witnessed significant progress in elucidating the molecular and cellular mechanisms underlying auxin action. High-resolution imaging techniques, transcriptomic and proteomic analyses, and genome editing tools such as CRISPR-Cas9 have enabled researchers to dissect the complex networks governing auxin biology. Studies in model organisms like Arabidopsis thaliana have revealed the roles of specific genes and proteins in auxin biosynthesis, transport, perception, and response. Comparative analyses across diverse plant species are shedding light on the evolutionary conservation and divergence of auxin-regulated processes, offering insights into the adaptability of plant form and function. Despite these advances, many questions remain unanswered in auxin biology. The precise mechanisms by which auxin gradients are established and maintained at subcellular, tissue, and organ levels are subjects of ongoing investigation. The identification of novel auxin receptors, transporters, and downstream effectors continues to expand our understanding of the auxin signaling network. The integration of auxin signaling with other hormonal pathways and environmental signals represents a major challenge, given the complexity and redundancy inherent in plant regulatory systems. Furthermore, translating knowledge gained from model systems to crop species remains an important goal for sustainable agriculture and food security. In conclusion, auxin biology embodies the intricate interplay between chemical signals, gene regulation, and environmental adaptation that defines plant life. The study of auxin has not only advanced our understanding of fundamental biological processes, but has also provided practical tools for improving plant productivity and resilience. As research in this field continues to evolve, it promises to yield new strategies for harnessing the power of plant hormones to address the challenges of global agriculture and environmental change. The journey from the discovery of a mysterious growth-promoting substance to the molecular dissection of auxin signaling exemplifies the progress of plant science and the enduring fascination of the green world.